Hi everyone, in this lesson we are going to cover the very basics of genetics, mitosis, and meiosis. So let's first go over some definitions. A gene is going to be a unit of heredity that is passed on from parent to offspring. It could be a segment of DNA or RNA that is going to encode for a particular type of protein to be produced. Alleles are different forms of a gene. So we could see them right here. Here's a dominant allele. Here is a recessive one. So for particular traits, you could have different types of alleles that could result in different types of phenotypes, which are different types of physical characteristics. Usually for alleles, we use a capital letter for alleles that are going to be dominant, and lowercase letters for alleles that are going to be recessive. A gene pool is a set of genes in a population of a particular species. Okay, chromosome, that's basically DNA that is mixed with different types of proteins. So DNA isn't hanging around by itself in the nucleus. It would actually fall apart. But there are going to be molecules around it and some other molecules that are going to protect the DNA inside the nucleus. The function of chromosome is to carry genetic information. Chromosomes can be found in the nucleus of eukaryotic cells and in the nuclear region of prokaryotic cells. Remember that prokaryotes, for example, bacteria, don't have membrane-bound organelles, so they don't have a nucleus, but they still do contain genetic information. In this particular presentation, we are mainly concerned with eukaryotic cells and not so much about prokaryotic cells. Chromosomes become condensed right before cell division takes place. So we could see condensed chromosomes right here. Keep in mind that humans, in your somatic body cells, you have 46 chromosomes or 23 pairs of chromosomes. There are two forms of cells in your body. You have somatic cells, which are pretty much everything except sperm cells and egg cells. All cells in your body, for example, your epithelial cells, your skeletal muscle cells, they all divide by the process of mitosis. In the process of mitosis, you get two identical cells, each one with 46 chromosomes. Now your gametes, which are your sperm cells and your egg cells, only have 23 chromosomes. So not 23 pairs, but 23 chromosomes. And they divide by a different process called meiosis. Meiosis will result in 4 different cells that are going to be haploid, meaning that they don't come in pairs. There's only one set of them, unlike somatic cells. And it makes sense because if you have a sperm cell that has 23 chromosomes and an egg that has 23 chromosomes, when they combine to form a zygote, you will get 46. Again, over here, you could just see here's a chromosome over here. Within the chromosome, you basically have coiled DNA, and this is what you're going to find in the nucleus of eukaryotic cells. Okay, somatic cells are pretty much most of your body cells. They are diploid because they come in pairs. So you have 46 chromosomes, you got one set from your mom, and you got one set from your dad. That will make 46. These chromosomes are called homologous pairs, and homologous pairs are not identical and may carry different inherited alleles. So you might have an allele that codes for brown eyes and another one that codes for blue eyes. It's really important to know the difference between homologous pairs and sister chromatids before we talk about meiosis or mitosis. If you don't understand the difference between homologous pairs and sister chromatids, you will be very confused. So I'm going to go ahead and just make a drawing over here. So you have the chromosomes that get condensed. So you have one over here and another one here, because remember, they come in pairs. And then you might have another pair right here and another pair right here. This whole thing, this is called a homologous pair. These two are not going to be identical. They have alleles that probably code for different things. So you probably got this one from mom and this one from dad. Now, before the process of cell division, the cell will make identical copies of its own chromosomes. So when it makes identical copies, it will look something like this. These two over here, these are going to be called sister chromatids. And these are identical copies. So again, this one and this one over here are homologous pairs, and the ones that are attached to them are sister chromatids, which are identical copies. Here you could see what chromosomes in a haploid cell would look like. You only have one set of them. This is kind of what it's like in the eggs or the sperm cells. And in your somatic cells, you have pairs of them. So they will be called diploid. Here are some other definitions to keep in mind, and these are important to understand when you're doing genetic problems. Genotype is a set of genes that an organism carries. So genotype basically means the genes that you have. Phenotype are observable characteristics of an organism. This is basically how you look, how genes are expressed, what are your physical traits. And phenotypes can be influenced by your genes, but also how those genes are expressed can also be influenced by the environment. So first, we're going to talk about something called complete dominance. This is when one allele is fully dominant over the other one. So for the dominant allele, we're going to use a capital letter. For the recessive allele, we're going to use a lowercase letter. In the case of complete dominance, one allele is fully dominant over the other. So when you're doing genetic problems, the first thing you should look for is what type of dominance they're talking about. Are they talking about complete dominance, incomplete dominance, codominance, X-linked dominance? You would get different results depending on the type of dominance that they are talking about. So that's something to pay attention to before you solve the problem. So here's an example over here. Remember, we're going to have two alleles because in your cells, you have diploid cells. They come in pairs. So you're going to have two alleles. If you have two alleles that are going to be dominant, we say that they are homozygous dominant. And in this case, it would be purple coloration in flowers. If you have two recessive alleles, we say that it is homozygous recessive, and it will result in white. Now, if you have a heterozygous, that means one allele is dominant and the other one is recessive, you're going to get purple coloration. And the reason for that is because this dominant allele, will basically override the recessive allele. So this is the one that's going to get expressed. Now here's something to remember. These two things over here are phenotypically the same thing. Both of them are expressed as purple. However, genotypically, they are not the same thing. You have two alleles here that are dominant, and over here you have one that is dominant and one that is recessive. So in a problem, when they're asking you about the genotypic ratio versus the phenotypic ratio, that's not necessarily the same thing, not always. So let's look at this problem over here. In sweet peas, the allele for purple flower is dominant to the allele for white flower. So right here, this is the first thing we should look at. We're looking at a problem that is talking about complete dominance. So if a heterozygous purple flower plant is crossed with a white flower plant. So let's go ahead and fill out the square. So if you have a heterozygous purple flowered plant, you're going to have capital letter P and a small p. And that is crossed with a white flower. And because this is the case of complete dominance, the only way you could have white is to have homozygous recessive. What is the potential genotype and what are the potential phenotypes? So let's go ahead and cross them. So in this case, you get a 1-to-1 ratio. 50% will be purple and 50% will be white. Again, in this case, the genotype is the same. 50% are going to be heterozygous purple, and 50% are going to be homozygous recessive. Okay, tongue rolling gene. So the tongue rolling ability may be due to a single gene, and those who are able to roll their tongue have the dominant trait, and those who cannot roll their tongue have the recessive trait. So let's look at this problem over here. If a heterozygous tongue rolling male mates with a non-tongue rolling female, what are the potential genotype of the offspring and what are the potential phenotype of the offspring? So we have heterozygous tongue rolling male. So a dominant allele and a recessive. Remember, in the previous slide, based on the previous slide, we already know that this is the case of complete dominance. And non-tongue rolling female, and the only way to be a non-tongue rolling female is to have homozygous recessive alleles. So if we cross them, we could see that 50% of their offspring will have the tongue rolling ability, 50% will not. Or in other words, they have a 1-to-1 ratio. Okay, now there is another type of dominance called incomplete dominance. This is a form of intermediate inheritance. This is when one allele isn't fully dominant over the other, and this is why it's so important to read your questions carefully. So what gets expressed when you have the two different forms of alleles combining is some sort of intermediate expression. So you have purple over here, white over here, and you get pink flower when you cross them. Only in the case of heterozygote organisms. Okay, let's go ahead and look at this problem. If tail length in cats is an incompletely dominant trait, what would the resulting offspring of a cross between a long-tailed cat with a short-tailed cat be? So here you have the long-tailed male, and this was a mistake, by the way. Short-tailed would look like this. So press this out. So what you're going to get over here is 100% offspring that are going to have medium length tails. Again, this is an intermediate form of expression, so the ratio would just be 1. Okay, looking at this one, if coloration in a particular flower is incomplete dominant, what would the cross between a pink and a red flower be? So here we could see a red flower and a pink flower. Now here you could see that there's going to be a 1-to-1 ratio. So 50% are going to be red and 50% are going to be pink. There's another form of dominance called codominance, and this is when the two alleles are equally dominant. So co means together. As you could see here with this flower, both the pink and the white were expressed. So instead of one trait being dominant over the other, both traits appear. This picture over here, this is really good for seeing the difference between incomplete dominance and codominance. So if you have a case of incomplete dominance, you would get an incomplete expression or intermediate expression of these two, which is pink. If you have codominance, you would get something like this one, where they're both going to be equally dominant and both pretty much are going to be expressed equally. Okay, in some cattle, genes for brown hair, capital B, and for white hair are codominant. So these two alleles are going to be codominant, equally dominant. Cattle with both alleles have both brown and white hair. This condition is known as roan. What are the genotype and phenotypic ratios of offspring when a roan cow and a white bull mate, and also when a brown cow and a roan bull mate? So first one, we have roan cow. So in a roan cow, you would have both alleles. And also white bull. So the only way to be white is to have both of the alleles. So here, again, you would have the 1-to-1 ratio. 50% will be roan and 50% would be white. Now in this one, we have a brown cow and a roan bull. And here you would get 50% roan and 50% brown. So again, we just could say it's a 1-to-1 ratio or 50% chance. One of the codominant traits in humans is blood type. So for example, you could be type A, type B, type AB, or type O. These all have to do with different alleles. So let's go ahead and look at these. So if you take a look at these alleles, this is how the allele for type A is written. This is the one for how type B is written, and this is the one for O. So this is interesting because type A allele and type B, these two together are codominant. However, they are both fully dominant over the recessive allele right here, which codes for O. So if you are a type A, you could be either one of these two genotypes. Remember, we have diploid cells, so two alleles. You could have two dominant alleles for type A, or you could only have one and have one recessive over here. Either way, you're going to turn out to be type O. Same thing with type B. You could have both dominant alleles for B, or just one dominant allele and one recessive allele. And in both of these cases, you are going to be expressed as someone who has blood type B. So right here, what you're seeing is someone who's type A and carries a recessive allele. Someone who's type B and carries a recessive allele. Over here, or you could have two alleles that are going to be dominant and different. You could have a type A allele and type B, which are codominant, so they're both going to be expressed. You would get type AB blood, and the only way you could be type O is to have two recessive alleles. This is a recessive allele, so if there's a dominant allele anywhere here, the recessive allele is not going to get expressed. Okay, let's go ahead and do this problem. A man with AB blood type marries a woman with type A blood. What are the chances that their offspring will be type O? First of all, if anybody, either one has type AB, there is a 0% chance that their offspring will have type O. The only way is for both parents to be carrying the recessive allele. So let's take a look. Here is AB. And type marries a woman with type A. So type A, let's assume that she has the recessive allele. We're assuming that she has it. She doesn't have to be this type. She could be this. But we're just going to make that assumption here. Okay, as you can see, there's pretty much a 0% chance that their offspring will have type O. 25% are going to be homozygous type A. So this is a case where the genotype and the phenotype are not exactly the same thing. You could see that right here, 2 squares, 50% chance of getting type A. This either one results in type A. So I'm going to put 2 squares. 25% chance that there will be type AB, so 1, and 25% chance that it will be type B only. So 1 to 2 to 1 ratio. This is for the phenotype. Now, genotypically, it's all 25% because genotypically, this right here and this one over here are not the same thing. They're not exactly the same genes, even though they have the same expression. Okay, let's look at this problem. A man with type A heterozygous marries a woman who has type A heterozygous. Are there any chances that their offspring will have O? If they're both heterozygous, that means that they both are carrying the recessive allele, and yes, there is a chance. So if we cross them, we could see that here is homozygous dominant. Okay. So as you can see here, there is a 25% chance that their offspring can be type O. So if I'm writing ratios for the phenotype, remember, phenotype is not necessarily about the genes, it's about how things are expressed. You're going to have 3 squares that are type A. All of these result in type A blood, and 1 that is going to result in type O. Now, as far as the genotypic ratio goes, that's going to be different because this is not exactly the same thing as this, even though they have the same expression. You have 2 squares right here with heterozygous type A. These 2 are the same thing. 1 square here that is homozygous dominant type A, and 1 square that is homozygous recessive. Let's look at something now called X-linked. X-linked has to do with a trait, with a gene that is going to be located on the X chromosome. So females have 2 X chromosomes and males only have 1 X chromosome. So diseases that are linked to the X chromosome are more likely to affect men because they only have 1 X chromosome, 1 allele, and that one is going to be fully expressed. So in this problem, when it tells you something is X-linked, the first thing that you should do on the Punnett square is to do XX and XY. If you put XX and XX, that's incorrect. Always the first thing you should do is put XX and XY. I don't care which side you put them. We just want to make sure that you write out that there's going to be one with 2 X chromosomes and another one with 1 X chromosome. So if a black male cat and a female calico cat mate, what would be the genotypic ratio of their offspring and what would be the phenotypic ratio? Sorry about this typo over here. So calico, it's this coloration of the coding. Calico cats are mainly white and they have white, orange, and black spots on them. This is a trait that you usually see in female cats because they have 2 X chromosomes. It is very rare to see a male calico cat. So, so here, if a black male cat, so I'm going to put the allele right here, B, this is X-linked. So you don't put anything on Y. So when something is X-linked, we only are concerned with what's going on the X chromosome. And a female calico cat. So I'm going to put B for black and maybe W for white. This cat has both of these alleles. That's why this is kind of like codominance in a way. They're both going to be expressed here, and because the cat has 2 X chromosomes, both of these alleles are going to be expressed, the white and the black. So if we cross them, again, only get 50% chance of female, 50% chance of getting male. So the first thing you should do is this. And now we're going to cross them. You have 2 X's here. And again, do not put anything on the Y. This has all to do with the X. And as you can see, this is why male cats cannot be calico. Calico has to do with 2 chromosomes expressing their alleles, 2 X chromosomes. Males have only 1 X chromosome, so only 1 allele is going to get expressed. So the result is going to be 25% chance of getting a female black cat, 25% chance of getting a calico cat, female cat, 25% chance of getting a male cat that is black, and 25% chance of getting a female cat that is white. Now I'm briefly going to go over mitosis. This is not usually something that we cover in Biology 2402. I go over it because I feel like it will help with understanding meiosis better. So the cell cycle has two parts. Interphase, this is when the cell is resting, it's doing its job, and it's not dividing. And then mitosis, which is very basically division of the nucleus, forming two identical nuclei and having them get divided. It is not necessarily division of the cell. Here's this little diagram here. This shows you interphase, which can be divided into three parts, G1, S, and G2. And then here are the phases of mitosis. So that chart that we looked at isn't 100% accurate, but it helps us understand different phases. So within interphase, there's going to be these three phases over here. You have G1, which is when the cell is growing. The organelles are dividing, and within G1 there's going to be a phase called G0, and this is actually where the cell spends most of its life, even though it's not on that particular diagram. It's not growing or dividing, it is mainly at rest. S phase, S stands for synthesis of DNA, so this is where the genetic information of the cell gets duplicated. And G2, this is where more growth happens, but this is also where, this is the checkpoint phase. So the cell checks everything to make sure that it is ready for division to take place. Here are the four phases of mitosis: prophase, metaphase, anaphase, and telophase. Do understand, have a good idea of what's happening in each phase, because that will help you understand meiosis better. Now, in some books, they will talk about things like prometaphase, early anaphase, late anaphase, early telophase, and that is because this is really more like a spectrum, but we just want to get the basic idea of what is happening under each phase of mitosis. So prophase, this is the first phase of mitosis. What you're going to see is that the nuclear envelope is going to begin to disappear, and the chromosomes are going to start condensing. And this condensing can really easily be seen under a light microscope. So you're going to see these dark lines forming in the nucleus of the cell. And centrioles, which guide the cell with division, move to opposite poles of the cell. So we see something like this. This is actually a plant cell, not an animal cell, but it's really nice to see different phases of mitosis with plant cells. You could see them much better than you do with an animal cell. So I used this slide. As you can see here, here's the nucleus, and you could see the chromosomes condensing. Here it is. This is a fish blastula, and it's condensing over here. And this is what it would look like on a model. Early prophase, and this is like later prophase, but we're not so concerned with that for Biology 2402. Metaphase, this is where you're going to have the nuclear envelope completely disappeared, so you're not going to see a nucleus at this point. The chromosomes are going to be fully condensed, and they're going to line up in the middle of the cell. And you're going to see that the centrioles have completely moved to opposite poles. So here are metaphase right here. This is again the fish cells, and the chromosomes are lined up in the middle. This is a nicer picture right here. And here's one over here with the plant cell. And right here with the model, these are sister chromatids over here. Anaphase is when the sister chromatids are going to get pulled apart. So you have identical copies being pulled apart. Here you could see it in a plant cell, and here it is in an animal cell, and right here in a model. Telophase is where you're going to see two separate nuclei beginning to appear. This cell might not be completely divided at this point, but you're going to still see two separate nucleuses forming. And once they form, the chromosomes that were condensed are going to begin to uncoil. So right here, two separate nuclei starting to form. And you can see the cell is cleaving off here. And again, here is late telophase, two separate nuclei. And right here. So this is like early telophase and late telophase, where the nuclear envelope is beginning to appear again. If you look at a plant cell under the microscope, it's really nice to see and really easy to see some of these phases. So here's like early phase of anaphase over here. Here is telophase, right here, the end of telophase. Cytokinesis is basically the division of the cytoplasm. Again, mitosis is not necessarily the division of the two cells, but division of the two nuclei, formation and division of the two nuclei. So cytokinesis is basically the pinching off of the cytoplasm, so you could have two separate but identical cells. Cytokinesis does not happen in plant cells, it only happens in animal cells. Plant cells, because they have a cell wall tends to form in between the two nuclei. Okay, so now we are going to talk about meiosis. So keep in mind that meiosis is very similar to mitosis, but it's kind of like mitosis happening twice. So you're going to have meiosis 1 and meiosis 2. So within meiosis, first we're going to have a prophase I, and this is important when you're taking a test or a practical. You can't just say prophase when it comes to meiosis. You have to be specific about if it is prophase I or prophase II. What you will see in prophase I, again, just like it is in mitosis, the chromosomes are going to begin to condense. However, here's the difference. Chromosomes pair with homologous pairs. So you have sister chromatids over here. You see the two pink ones that are attached to each other. These are sister chromatids. But remember, your chromosomes come in pairs, so it's also with the homologous pair. This forms something called a tetrad. So you have 4 of them right here, 4 of these long structures. And a process called crossing over is going to happen. So there's going to be an exchange of genes happening between the chromosomes. So like you would see right here. So these two together are homologous pairs. And each one has made an identical copy of itself. So right here you have these two sister chromatids and these two sister chromatids. They're going to come together to form a tetrad. And as you can see, they're basically going to exchange some genetic information. This is important for creating genetic variety in your gametes. So this is why you and your siblings don't look identical. You have this recombination of genetic information happening in the gametes. Keep in mind, crossing over, it's going to happen in prophase I of meiosis I. Once prophase I has happened, and you could see here, now you have chromosomes that are no longer really identical to each other. Even the sister chromatids can't be considered identical in this case because crossing over happened. Homologous pairs, not sister chromatids. So here we're more concerned with homologous pairs in Metaphase I. Homologous pairs are going to be lined up in the middle. So now you have 4 of them over here. In Anaphase I, homologous pairs are going to get pulled apart. Be careful, sister chromatids are still attached to each other at this point. So Anaphase I is where homologous pairs are going to get pulled apart. And then Telophase I, you're going to have two separate nuclei forming over here, so you have two cells. Kind of like mitosis. Now, we want haploid cells. We don't want diploid cells for our gametes, so the process of meiosis is going to happen one more time. So now we have two cells, and they're all going to be slightly different because crossing over has happened. So what's going to happen here in Prophase II is that the nuclear envelope is going to disappear again. The chromosomes are going to condense. These centrioles are going to start to move to opposite poles of the cell. But this is Prophase II. So now you have sister chromatids, no longer homologous pairs. Sister chromatids lined up in the middle, in the midline of the cell. Here are the centrioles, and you could see the esters right here, these fibers that are going to extend to the sister chromatids to help pull them apart. In Anaphase II, now you have sister chromatids pulled apart. And again, because crossing over happened, the sister chromatids are not identical, so you're not forming identical cells. And in Telophase II, now you're going to have 4 different cells with nuclei that contain different types of genetic information because of crossing over. So the result is going to be 4 haploid cells. Something to keep in mind is that in the process of meiosis for males, all 4 of these cells will become sperm cells. Males are not, they don't have a restriction in terms of how many sperm cells they produce. They're constantly producing sperm cells. However, females are born with a set number of eggs. And those eggs, from birth to all the way through puberty, are going to be arrested at Prophase I. So they're going to remain at Prophase I of Meiosis I. And during each menstrual cycle, Meiosis I is going to be completed for an egg cell. And during ovulation, the process of Meiosis II begins for the egg cells. However, if the egg does not get fertilized, it will not make it any past Metaphase II. So it needs to be fertilized for the process of meiosis to be completed. If it gets fertilized, it will quickly complete the process of meiosis. And keep in mind that for females, only one of them will be an egg. The rest will become something called polar bodies.